All right, so welcome everybody. It's the evening monthly program of Los Angeles Audubon Society for February 2023. My name is Travis Longcore. I'm president of LA Audubon. I want to welcome everybody to the talk tonight. We're here and streaming live over on Facebook. And so I did want to let you know that if you found out about this meeting from some source other than our mailing list and you'd like to be on our mailing list, you can go to laaudubon.org. Down at the bottom, there's a link about every page. There's a button to push, and we have a number of different mailing lists for evening programs, for the Rare Bird Alert, for other events that we have, bird walks, etc. And so I welcome you to join up there. Also to keep an eye on the calendar at laaudubon.org for events, including open wetlands twice a month at the Bayona Wetlands, bird walks, and the occasional longer field trip. We've just scheduled one to the Dancing Chicken up to see some DWP property up near Owens Valley coming up later this spring. So go ahead and keep an eye on those things if you're interested in those events. So tonight we're really delighted to have a longtime collaborator of LA Audubon, Tom Ryan, to talk to us. He's a consultant here in Los Angeles, does some teaching as well, is finishing up his PhD, and has a great presentation and special guest prepared for us tonight. It's the conservation of the least tern, a bird without borders. So with that, I want to welcome everybody again, and Tom, take it away.
All right, Travis. Well, thank you very much, and thanks everybody for coming out tonight. It's been a number of years I've been working on this, and really what I want to do tonight is share with you, not just a little bit of information about the least terns, but also really kind of bring you into the project we've been working on, because I think it's one of the most special things I've ever been a part of. And it's one of those rare situations where, 1 + 1 = 3. And we have gathered just an amazing team of people together, a great group of collaborators. And, I feel like we're really making a difference, not just in our knowledge of the bird, but also what will hopefully be the long-term conservation of, not just the least tern, but also estuaries and beach habitats throughout California, the Baja Peninsula, and hopefully mainland Mexico. So let me go ahead and bring up the presentation and get us going here. So this is our wonderful little creature. There we go. So this is our wonderful little creature, California least tern, the least tern, the Gallito marino, charan minimo, or the scientific name Sternula antiarum. The way that this project kind of got started and got going was a photograph. So my good friend and current collaborator, Jonathan Vargas, was down in the estuary at La Paz, and he got these two pictures of these two different individuals that both had bands on them. The one on the, would be my left, has the white band above the tarsus, the orange band below, which is, a pattern that nobody knew who put that on. And then the one on the right is very clearly a metal band, very similar to the one from the U.S. Fish and Wildlife Service. So the only bands in the bird banding labs registry had been put on in California. Nobody had ever submitted data that bands had been put on the Baja Peninsula. So we immediately thought, okay, well, maybe we've got a California bird at a breeding colony in La Paz. That's pretty interesting. So we went down and looked for it, and turns out that wasn't quite the case. Many of you probably know Charlie Collins, and Charlie had worked with Eduardo and another one of our collaborators. And back in 2003, 2004, they'd put a whole bunch of bans on these guys. So it was one of those locally applied birds, but still extremely interesting. So that led to a whole series of meetings among biologists on the peninsula. And so, we began to put together this team. And on that trip down to La Paz, I met both Edgar Amador and Graciela over on the left there. I'd known Liliana and Jonathan for a long time. Same thing with Eduardo Palacios. There's a picture of me at like 21 with Eduardo. And we decided we were going to try to put together a team to actually study what was happening. Edgar had been working for many years on the peninsula, pretty much by himself. And we wanted to put together a team to figure out what was going on in the Baja Peninsula, and then also to see if there is any connectivity between the peninsula and California. That was kind of how this whole thing got started. And so went to Mexico and started doing training sessions. We worked with groups. This is a training session for the group in Los Cabos, and had a great group of people there, including some very young collaborators. And Ensenada, we worked with both Terra Peninsular and Proesteros to set up a group working there. Liliana had already been working for quite a few years, so that made it really easy to set up. And then in our really after our first full year, we were blessed by having this Pacific Seabird Conference occur in February 2018 in La Paz. And we brought in a whole bunch of the students, as well as biologists from Chile and all over Mexico and the United States. And we got everybody together. We rented a kind of a hacienda. Everybody stayed there. We turned the Ramada into a discotheque 2 nights, which was a lot of fun, and it really kind of sealed the relationships. And we were able to really get the program rolling, and the enthusiasm for that has carried right up until this year, and everybody's getting ready for next field season. And what we ended up with was a monitoring network out on the Baja Peninsula. So we have a group of people who, it's a lot easier to work with the least terns there than it is here. But just a group of community volunteers that go out. They're headed by Graciela, who's a professor and actually a herpetologist. But, that's easy enough. Let's see. Something's going on with the presentation. Is everything okay?
Yeah, I think that you got knocked off the screen share. So if folks can be sure not to hit the screen share button, you just need to reshare your presentation.
Gotcha.
There
is it up again?
Yes, it is.
All right, great. Okay, and so yeah, they started working, and we've had just a tremendous amount of success, and that kind of leads us. I'm going to just very quickly turn this presentation over to one of my collaborators, Celine, who has joined us in the last year and has really started to document the connections that the birds make among the people. So Celine, go ahead and put your mic on. I'm going to turn mine off here, and let's see.
Great. Thank you, Tom. Awesome. And thank you for including me in today's A Bird Without Borders presentation. I'm very happy to be able to contribute a different angle of least tern conservation. I am a freelance photographer based in Orange County, California. And while my background is primarily working with brands in commercial photography, I am passionate about environmental stories. I hope to spend the next 10 minutes showing you how my experience with Least Terns has transformed my photographer's identity to include conservation photography. Next slide, please. If I had to choose one word to describe my first field season, it would be opportunity. I have learned that, like the birds and the people behind the birds, I am an opportunist. My project, Least Resistance, came from taking a risk and making myself available during the nesting season. As I began to follow the birds, I started to see a bigger picture. While it may be dispersed, as Tom was just explaining, there is a Least Tern community made of committed individuals, groups, and organizations. Regardless of their geographical classification, as California Least Terns or Mexican Least Terns, these birds have become meaningful connectors of people, places, and culture. Next slide, please. So last year, I was able to visit 20 unique nest sites in the United States and Mexico. These birds' protected status, resilience, and migratory nature give Least Terns the unique ability to break through social and cultural barriers and create opportunities for people to work together between two countries. As a hybrid of science and storytelling, Least Resistance is about tying together the stories of the people behind the birds along their migratory route in order to highlight the benefits of building meaningful connections to our shared environments. Next slide, please. So before I entered into the field, I volunteered at Bolsa Chica Ecological Reserve, and it's through the Eyes on Nest Site program, and Jeanette Bush, who I saw is on this call, which is where I first learned and was introduced to the endangered California Least Tern. As a monitor, I learned to spend hours observing their behavior, to appreciate their liveliness, to understand why people as protectors are necessary, and to feel like I was participating in their protection. However, when they left, I had an obvious question. Where do they go? Which then led to, are they still protected wherever they go next? And if so, by who? Next slide, please. This project would not have been possible without the scientific partnership of Tom Ryan, who took a chance and a risk answering some of my questions and encouraging a lot more. I'm very thankful for his invitation to document his ambitious research study of Least Terns in the Gulf of California. I'll never forget the excitement I felt receiving the detailed trip itinerary, and one of the takeaways from a preseason trip to Guerrero Negro, which was, there's a lot of good science being done, but not a lot of public knowledge about it. This is where I believe visual storytelling can make a difference. Next slide, please. Entering into the field was an adventure. Very long car rides, questionable sandy roads, vehicles stuck in sand, and the possibility that we wouldn't find any Least Terns. For me, it was an introduction into field work that allowed the visual exploration of three broader themes. First, learning who the people behind the birds are, how they are organized, and listening to why they do what they do. Second, identifying the many layers of connectivity. Whether that means between California Least Terns and Mexican Least Terns, the environment shared by Least Terns and people, or the people in two countries working to protect the same bird. And lastly, understanding our relationship with nature and how least terns are transformative in that they create emotional ties to places. While none of these themes can possibly be done just in one season, I'd like to use the next few slides to show some of my learnings. Next slide, please. I'm not sure if it ended up moving. Next slide. Perfect, thank you. So the first lesson, the people behind the birds are experts like Tom, but they are also students like those part of the Baldwin Hills Greenhouse Program. At Bolsa Chica Ecological Reserve, they are California Department of Fish and Wildlife, Bolsa Chica Conservancy staff, and Eyes on Nest Site volunteers working side by side to remove vegetation during the off-season. Next slide, please. Maybe there's a lag.
Change freeze line.
I think it's lagging because I still see the previous one with the students.
You know, we're all seeing one with multiple pictures, including what looks to be a salt or a sand dune.
It could be, it must be on my end.
Yeah.
Actually, let's go back to the one with quotes from Stacy and Graciela.
Okay.
Great.
Yes, the one with the quotes is up now.
Perfect. Thank you. So the second lesson is, while the word conservation aligns all of these efforts, I found that it means something different depending on the characteristics of the environment. For example, while the Venice Nest Site provides Los Angeles residents an opportunity to experience natural beach habitat, the efforts in Los Cabos are tailored to educating hotels and tourists whose hotel zone is also nesting habitat, and including them in seasonal protection. Both of these tern sites exist in highly developed areas, unlike the coasts of Sonora and Sinaloa, which means building empathy for the land people populate the most is extremely important and is often missing. In remote areas such as Punta Banda, conservation is about making the land more accessible to people involved in monitoring least terns. And now the next one with the series of, perfect. Thank you. Lesson 3. Before entering into the field and based solely on researching different nest sites, it was clear that least terns coexist with a wide range of human activities. These birds are resilient. They have created temporary homes near airports, salt mines, oil fields, the military, popular tourist destinations, and one of the world's busiest seaports, just to name a few. While all of these people have least terns in common, it wasn't until I began reviewing images of their environments that the subtle repetition of certain textures, colors, shapes, reinforced that these seemingly disconnected environments are in fact all a continuation of shared land, just with a different sense of style. Next slide, please. Lesson 4 was I was pleasantly surprised to learn that part of Tom's methodology involves photography, which shows how photography is such a versatile and important tool for conservation. As a conservation photographer, my job is to help translate the science and to create visual experiences that both support the work that is being done in the field and complements it by bringing the subject into spaces that it can't exist in without photography. Next slide, please. As the 2023 Least Tern season approaches, I want to reiterate that I view this project as a longitudinal documentary story, which means something I hope to continue for many seasons. There is a lot left to be discovered. How will this community change over time? Who else is part of this narrative? Will we ever learn where they go to winter? My experience so far has revealed many challenges, including what do I do with all of these images? However, what inspires me to continue this project is seeing how it is the birds who are teaching us how to overcome our differences. Next slide, please. I'll end with my first step. As part of my figuring out what to do with all of the images process, I created a traveling pop-up exhibit. My hope in hosting future pop-up exhibits is to create an occasion for people to meet each other in person and to experience the power of the Least Tern community. Next slide. Thank you.
All right. Thank you very much, Celine. And on our end, I can't tell you how important it is. One thing that scientists routinely do is write our findings in these, very, technical, kind of difficult to read papers. And then, the wonder and the amazing experiences of the field work just kind of get lost in that. They end up in stories being told at meetings, and it's a real pleasure to have Celine around. And also you're going to benefit very much this evening from her photographic skills as opposed to mine, which leave a bit to be desired. So thank you very much, Celine. And that really introduces kind of the rest of the talk. This is these are the band combinations that we use. It's not mistake, not by any accident, the colors of the Mexican flag. That was one of our collaborators that chose the red to go with our green and white from the States. But we've created this, again, the training has paid off. We now have people throughout the peninsula that are knowledgeable in the ability to capture, handle, and mark these Least Terns. So this is our team down in Los Cabos. Here you've got a chick being marked with a alphanumeric field readable band. And then, the goal is, and this is one from California, it's got a white alphanumeric code, is that we can get photographs of these at the nests using a variety of photographic methods. But the main one was actually innovated there in Mexico. So this is a picture of Miguel. He's one of our biologists / volunteers down in Los Cabos. And he had a GoPro and decided one day to go put it right by the nest and see if he could get an image of the bird. And lo and behold, he was able to get a picture not only of the bird, but a very clear picture of the band. And so we immediately went out and bought a bunch of these, and we've been using them ever since. Other innovations are the crew. This is the crew in La Ribera, improving upon my box trap with locally sourced materials. And it's really been amazing. So we built this network of independent researchers and volunteers. And the peninsula has been a great place to work. The combination of all the people working together has done a tremendous amount to increase public awareness. They've done a great job of working with government officials, and the attention begets protection, and especially in Mexico, and in the tourist area around Los Cabos, just making people aware of this has really helped. It's increased the protection for the colonies, and we've been doing really, really amazing science. So, in addition to the public outreach, turning our volunteers loose, it really it's just a matter of how many bands I can get them. We estimate the population down there probably is somewhere around 700. And of those, they have marked, a large proportion. There is a good chance if you take a picture of a least tern in Los Cabos, La Paz area, it's going to have one of our bands on it. And with that has come 376 recaptures. And what we have found in the southern part of the Baja Peninsula is that there's interchange among La Paz, La Ribera, Los Cabos, and Cabo San Lucas. But none of the birds that we've marked in the southern peninsula have shown up in the northern part of the peninsula or the mainland. So that has been fantastic. So, again, we're continuing to collect these data, but it is giving us information about, in the strong indication that those colonies out on the southern tip of the peninsula may be an isolated group of nesting birds. In contrast, what we have found at Estero Punta Banda is that there is a tremendous amount of interchange between the colonies in northern Mexico along the Pacific coast at Punta Banda, which is near Ensenada, and San Quintin with all of our Southern California colonies. You can see a lot of the birds, and part of this is a function that the numbers are in part a function of where the bands were put on. Most of the bands in Southern California have been put on at Camp Pendleton and Naval Base Coronado. But we do see a lot of interchange, more than would be proportional with the Tijuana River estuary. So there is a lot of birds that go back and forth across the border, and when things are good in one country, they go there, and then they move to the other country. Now, one of the real interesting returns we've had is that last one. So this is a chick that was banded in 2019 in San Quintin, which is in the northern Baja Peninsula, but pretty far down. And it was observed, again, all you need to recapture a bird is a photograph, at Oceano Dunes, and that's 420 miles north. Now, just to put this in perspective, and this becomes really important later in the talk, the distance from San Quintin to Oceano Dunes is pretty much the same distance from San Quintin to one of our southern sites in southern Sonora at Estero Tobari. And that distance encompasses, this whole coast of the Upper Gulf, Guerrero Negro. The question, are the birds capable of dispersing to other places? Absolutely. And this kind of becomes interesting. One more, and then I'll tell you why it becomes interesting. And then this is another observation. This is one of our alphanumeric birds. We couldn't get the read on the actual combination, but the fact that the band's a little bit bigger than the metal band clearly makes this one of our Southern California marked birds. And this bird showed up in amongst a group of 241 migratory least terns in the Upper Gulf in July. So it does look like the least terns here in California likely move into the Upper Gulf to begin their migration. And then there's some other corroborating evidence of banded birds being seen a little bit farther south as well. So, the movements are real interesting. Now, that begs the question, of course, is where do least terns occur? And it was kind of fun. I went online and just looked, Googled it. And this is one of the maps that comes up. And this is a horrible map because it shows the terns, through to about, Puerto Vallarta. But we know that least terns nest all the way down into northern Costa Rica. So this map is very inaccurate and dated. And then I found this one too, which was rather surprising because if you look at all of the papers that have been done on relationships of least tern subspecies, it says that the coastal population of Mexico is the same one as the Gulf Coast, and there's absolutely zero evidence to that. So Right now, probably the best source for least tern distribution is actually eBird. I check it regularly for pictures, hoping to get one with a banded bird. But the least tern nests pretty much all the way from San Francisco down into Panama and into northern Costa Rica on the west coast. Wintering range is pretty much from about the area around San Blas, Puerto Vallarta, south through to Panama, but there aren't that many sightings. Some of the more reliable sites are right around Puerto Vallarta at what's called the Banderas upwelling. And then the other one is down on the Isthmus of Tehuantepec. Whoop, there's his. Near on the coast of Oaxaca, near some really large salt ponds, near a large upwelling here. We don't know where they go, but one of the things is you have breeding colonies all the way down the coast. So these birds that are being seen on the coast of Mexico, they could be migrants from California, but they also could be local breeders. So it makes determining migration very, very difficult. And taxonomically, this is a bit of a nightmare as well. So there's kind of 3 currently accepted subspecies. You've got the California least tern, the interior least tern in the river systems, and then the East Coast least tern, which goes down the East Coast, the Gulf, and then out into the Caribbean. There are papers describing the Mexican least tern, quote unquote, occurring from the upper Gulf down to, well, Colima. Again, this map's kind of a mess. And then another one called Stableri, kind of the Central American least tern, along the coasts of Guerrero, Oaxaca, Chiapas, and down into Central America. Now, the descriptions of both these Mexicanus and Stableri are iffy at best. There are some differences that they describe that actually can be measured, and I'll get to those in a little bit. But basically, kind of looking at this in terms of our colonies, these are the red dots are the colony sites. This is the California least tern. These are likely California least terns. Sometimes these guys are considered California least terns, sometimes not. And then this is the Mexican least tern going down the coast of Sonora and Sinaloa. Now, these birds live under very different oceanographic conditions. And when we start looking at, are they connected? Are they related? You have to look at the oceanographic conditions and kind of, how do the terns see the world? So all of these guys out on the Pacific coast, at least through to Guerrero Negro, are under the influence of the California current and the upwelling systems associated with this current. Very cold water, probably very similar prey base up and down this area. Up in the upper Gulf, it's a very, very different world. The upwellings here are actually driven by tidal movements. So these birds are dealing with very different conditions. Lower Gulf has a lot to do with the winds, and upwellings change from one side to the other depending on the winds. And then these birds down in the southern part of our study area are under both the influence of the southern Gulf and the upwellings of the winds here, but also the Mexican countercurrent. So, while, we're looking at birds that look very similar, they live under very different conditions. One of the other problems we currently have is kind of the concentration of studies. So most of the money and work that's done is done in this little red polygon up here in Southern California. Okay. Unfortunately, this is a bird that, ranges, all the way down. And so while you have hundreds of biologists working on this bird in Southern California, and that 500,000 a year is probably, well above that, the whole rest of the range pretty much operates on a budget of about $10,000 a year. And most of the people who are working down here are purely doing it on a volunteer basis and just out of love of the species. So, from San Quintin to La Paz, there's 2 people. From Mazatlan to Costa Rica, there's nobody that regularly works with least terns at will. So it's a rough situation. Now, one of the problems is in that little red polygon, this is our current situation. This is from the most recent statewide data. And you can see the black line are the minimum maximum numbers of breeding pairs. This is a population count, and it is declining. There's been a lot of problems, in particular with predation, problems with protection of colonies, and then what appears to be the big one is the shift that occurred out in the Southern California Bight in 2008. And ever since then, we've had pretty large chick die-offs that have occurred throughout the years at some point during the nesting season. And this is probably due to problems with the prey base here in the Southern California Bight. The other problem is that, looking at just the volunteer counts, the numbers are, from 2016 to 19 estimated almost 4,000. When we then did a mark recapture using our bands, the estimate was considerably lower. And one of the things that we've observed is that the birds will visit multiple colonies if they fail in a given year. So in these years, as things get rougher, they fail more often, and we're actually double and possibly even triple counting them at other colonies. So one of the big problems is, if they're not here, there's two options. One, they're dead. That's bad. The other is that they went somewhere else. And least terns are prone to do that. So, with that problem in mind, I went back to school. My dog doesn't care. And started this project. So this is my dissertation. I'm working out of Cesese and Ensenada with Eduardo Palacios. And there, these are my stated objectives. And one of the big ones is, population connectivity. Is there regular or occasional exchange between the nesting colonies in California, Baja California, and we also want to create a population model for Southern California. We're continuing to collect data since that mark recapture population that came out. That we've got 2 more years of data. And then the other question is, as Celine alluded to, we really need to find out where these birds are going. So, the numbers are declining in our area, but, how much of that is what's going on here, and how much of it could be something that's going on someplace else that we're completely unaware of. So we need to find out where these birds go the other 8 months of the year. So the plan is, last year and next year, we're going to be visiting the colonies all throughout the Gulf and the peninsula, trapping birds, collecting feather samples, trying to figure out migration. And then the next step also is to look and see if these populations, are genetically connected, or if there are clusters, if there are groups that are isolated, particularly in the upper Gulf and the southern tip of the Baja Peninsula. These are both areas that are under tremendous pressure from recreation in the upper Gulf and then from development down in the tip of the peninsula. So I stole Celine. I love this map, Celine, so thank you very much. Most of the pictures from here on in, by the way, are hers. So we took off, and our first objective was just a little road trip down to southern Sonora. We started in the area of Ciudad Obregón and then worked our way north. We worked our way through Guaymas, Quino Bay, up to the area around San Jorge, Puerto Penasco, Golfo de Santa Clara, and then later on made a second trip down into Sinaloa to Ceuta. Meanwhile, we had other people working both up in Estero Punta Banda and the tip of Baja last year. So southern Sonora was a bit of a road trip, shall we say. A lot of miles, a lot of highway. And we got down and we met up with a biologist, Herman, and he took us out to this really unique area. We had to go out to the island by boat. It's a dredge island. There's a number of them out in this bay. And it turns out, Tobari, this is where the type specimen for Mexicanus was collected. So going back to where the first one was originally collected in 37. And we got out there. This is us. And, when we're out there, we just set up, little station with all the equipment and then take the traps out and start trapping the birds. So this is my very high-tech drop trap. It's a kite string, a stick, and a cage, and we just put it right over the eggs and catch the birds. So they they think it's kind of interesting looking, and typically they go in. It takes about 5 minutes or so for most. And once they're captured, we're putting these bands on them. We take measurements. So here's the process of banding them. That's a chick, but wing roll. We weigh them with Pistola scales. This is Herman. He's the biologist. He actually is a scientist at an agricultural school and has a strong interest in wildlife and has been helping Eduardo with his snowy plover studies. And then these are our boatmen, but these guys also just really, enjoyed seeing us out there and enjoyed talking to us, learning, and really got a kick out of showing us the dolphins that live on their route and one of the other islands. It was a great experience to be out there with them. And again, these guys are just, Herman does this on a volunteer basis, and the people that help him are just fantastic. One of the things that we were doing was collecting feather samples for stable isotope analyses. So we were taking about a centimeter square from the first primary, the third primary, fifth primary, 7th, 9th, and 10th, and then we'd alternate wings. So it would be just 3 samples from one wing and 4 from the other. Now with stable isotopes, the idea of this is that what we're hoping is that we can learn a little bit about their movement. There is a chance we might be able to get lucky and get geographic information, not terribly precise, but at least give a region as to where they're going. But kind of more important for our study, one of the things that we're definitely going to be able to determine is if these birds all move in the same manner during the non-breeding season. So we'll be able to look for patterns and make comparisons among these different colonies to see if these birds are migrating together, or if not only are they breeding separately, but if they're moving around separately in the winter. And of course, lots of camera data. Every once in a while, the terns do get curious about the camera, but just to give you an idea, we did the quick analyses, and it takes about an average about 5 minutes for a tern from the time it lands to when it goes into the trap. With the cameras, it takes about a minute and a half from when the tern lands to when it sits on its nest. So the cameras are definitely a lot less impactful for the birds, and we can, as far as reading the bands go, we can get the same thing. Now, one of the other things we were doing with the birds is collecting DNA for an analysis of DNA. We used basically just little diabetic finger stick needles and got a little bubble of blood, and then used a micropipette to transfer it to a thing called an FTA card, where you can preserve the blood, and you don't need, back in the old days, you'd have to carry a liquid nitrogen canister with you. Once you get the blood onto the preservative paper, it's pretty much set. So that makes life a lot easier. And these are some of the pictures from central Sonora. So this is us again, right out on. We had to wade across the opening of the channel out to the island. There were these little bitey crabs that were biting our toes. And once we got out there, we were able to trap. But this colony, and one of the other things we're doing as we go is kind of assessing what the problems are. And this colony had just been hit by coyotes. And the number of nests had declined from the previous week, and there were tracks all over the place. So, again, that's one of the assessments. And this is what it looks like when we're out there working with the cameras. So the camera is just placed about 25 centimeters from the nest, and these are the types of images we get. One of the other things to kind of look at with these birds is kind of interesting. Note two black primary feathers on this guy. So this is from Cardinal. And this is our collaborator there, Jaime. These guys were from Keno Bay Research Center, Prescott College, and they do a whole water bird monitoring program there. And as part of that, they also count least terns. And we found that to be the case too. A lot of people who are out there were working on other research projects, but while they were out there, they were also collecting data on the terns. And the terns nest with Wilson's plovers and American oyster catcher, and as well as here with snowy plovers. And efforts to protect all of these species, any one of these species benefit the others. And so this is the crew. This is having coming back through the channel and getting back in at sunset. In northern Sonora, the colony, we worked at two colonies. One of them was San Jorge, and the other was at Estero Marua. And this was a lot more similar to some of the colonies we see in Southern California, where it's more of a dune habitat. Both sites had fine sand and sand dunes. This one got to be a little pebble plainy in some places, but this is San Jorge. And this gentleman here, he has been monitoring and protecting these terns for decades. And he submits his data to the local wildlife office every year. And he does it just because he loves them. Nobody pays him. He's out there. He's worked with a local fish camp to help minimize their disturbance of the terns. And again, we just ran across this again and again, where people were just out doing it just because they love the birds. And here's some of our shots from here, and you can see again, like Celine was saying, these sites have very different textures to them. Some it's more sandy, some it's more muddy. Here there was a lot more of a shell component. This is Estero Marua. This one was one of the sites that was more sandy and duney. Just great. So one of the questions we get is, when do we trap? When do we just get pictures? And so the trapping that was necessary in order to get the feather samples and the DNA samples. The cameras were more for attempting to detect whether we had any banded birds at these colonies. So we got a decent sample at a bunch of the colonies this year. We're going to be going back down, and that's going to be one of the goals, is to get more, a much larger sample, particularly at Estero Marua and San Jorge, which are the biggest colonies closest to Southern California. And this is, again, our collaborator at Marua, Karen Baker. Her and her group of people at their little residential community have been protecting this little area at the end of the peninsula. They've put up signage. She just has an interest in birds. And she took it over from the person who originally leased the land to the people to build the houses. And he had a really strong conservation ethic and had been the original one to put up the signage and the little kind of symbolic fencing around this colony. The next trip was kind of a long one. We flew down to Mazatlan and then drove up to Ceuta. Celine was a real trooper. We stayed in what is best described as a field flop house with the biological crew. So it was like Celine and five guys in this little 2-bedroom, thankfully 2-bathroom, little place. Conditions out there were just beautiful. This site, it's a big salt pan, and whereas in Sonora and Sinaloa, I had Liliana assisting me. and Liliana's a tern biologist. Out here, Celine not only was taking photographs, but she became my field assistant as well as we were working. Now, this picture, you might notice, we were, anytime we encountered dead least terns, we collected them. And these were transferred. They're currently at the museum at the university there in Ensenada. And we were able to, again, to get samples from the feather and DNA samples from these specimens. And so Ceuta, again, this is a salt pan. A lot of the nests were in just footprint depressions. It was really, really interesting. And after that, it was back home. And we were home for a few days and then back in the field down to Ensenada. And here we were really anxious because in 2021 we deployed 25 GPS units on these terns, and we were really hoping that we would be able to get them back. And Ensenada, that's where Liliana is based, Jonathan's based. This is one of my students from PCC, went down and helped us out. Just a beautiful place. We camped out, the second time we camped out on the peninsula. And in 2021, we deployed these GPS units on the least terns. And the hope was that we'd be able to recover them in 2022. So the least tern's only 44 grams. And you can't get a GPS yet that transmits. So if you deploy them, you have to recover them. And so this is what it looked like with the antenna and the GPS on it. And we returned in 2022, and we saw our birds, and none of them nested. So we are very hopeful that these guys are going to come back again in 2023, and we will get somebody actually on a nest. So the problem is we can only trap when they're on nests, and so if they don't nest, we can't trap them. We can't otherwise collect them. So we just have to be patient and hope for 2023. One of the things we think happened is numbers everywhere were down again this year, and we had a huge number of banded birds from San Diego. So I think that was part of the problem down there. So the question of where they go in the winter, you saw from the eBird that there's pretty much sightings from Puerto Vallarta all the way down into the Gulf of Panama during the winter. It is very likely that these birds are going into areas of upwelling. And based on some studies that we did earlier, we have a pretty strong feeling that once they leave land, they don't touch land again until they nest. So we think that the least terns become pelagic and likely concentrate off of the Banderas, Tehuantepec, Papagayo upwellings, and then also down in the Gulf of Panama. So that's what we think. We'll hopefully be able to confirm that next year. So until that time, this is Andewi, and she's the master's student who's working with us. And so this is the stable isotope lab down in Sisese. And so she just spent the better part of 2 months processing all of the feathers. So taking them, washing them, cleaning them, preparing them, weighing them. And we had I think almost 200 individual samples that she had to work. So they're all processed, and just all we need now is to get the results back. And then the other thing that we're doing, and actually I'm going over to the LA County Museum tomorrow with a bunch of my students from PCC, is we're doing museum work. So we're taking a look at all of these specimens that we have in collections here in the United States, which most of those are here in Southern California at UCLA, Moore Lab, LA County Museum, San Diego Museum of Natural History. And we're doing some basic morphometric measurements comparing Mexicanus and brown eye, so the Mexican and California Least Tern. Kind of cool. Here is the type specimen. So this is what Mexicanus was described from 1937. So that was kind of neat. But we're just taking all sorts of morphological measurements and just kind of good old-fashioned museum work. So this is what I showed the students, and just looking and seeing if there are any differences. And what we've done is we've gone back into the original descriptions, and one of the things was that the Mexicanus was smaller, and it had fewer black outer primaries, were two of the things that it was described from. So we've taken a look, and you can see that our samples from this are pretty small still. But kind of interestingly, these are preliminary results, so don't take this too far. But it does look like the Mexicanus wing length is a little bit shorter. And on average, the California least terns, the ones on the Pacific coast, have 2 to 3 black outer feathers, and the Mexicanus have between 1 and 3, with most of them having 2. So there are some plumage differences in these birds. No difference in weight, but this wing is kind of interesting as well, because often one of the patterns we see in nature is birds that that have a migratory population. The wing length of the migratory population is longer than the non-migratory population. So one possibility is that these birds that people spot on the coast of West Mexico are from the local colonies, and brown eye is actually more of a migratory one that may go farther out to sea, or may join black terns down in the Gulf of Panama. So again, that's speculation. These are data though, and so far we are seeing some differences. So we got into a lot of great adventures when we were down there. Mexico is a wonderful place to travel around. Even with the Land Cruiser, we got stuck, I think, 4 times this year. We got out. It was a lot of fun. The food is just amazing in Mexico, including some very creative dishes. This was served at one of the meetings, and just a little bit too accurate when it came to the birds. And we, again, we're out in the field for long hours. Galipollo became our mascot. So these are carried around, and if you screw up in the field, you show up late, you've got to carry Galipollo around your neck for the rest of the day. And Galipollo got into all sorts of trouble during the trip, bit of a drunk. But we just had a really good time, and we're getting good data. We're working with just amazing people on both sides of the border. And I really kind of hope I've brought a little bit of, just the wonder of this whole experience to you guys. A little bit of an understanding of the process. You're getting me right in the middle of a study. So I'm a little short on results, but I do hope to come back and talk to you guys once we get the findings. But in the meantime, there is, that question of, what can I do to help? And so we've created a wanted poster. This goes out each year in both English and Spanish to a bunch of different listservs. One thing with the Mexican biologists, they give each bird a name. So in addition to the alphanumeric code, they also have a name. So everybody has an individual name. And so if you do see one of these bands, please do report it. I'm working with Travis Wooten, who many of you guys know from the Snowy Plovers. And he checks the Google Groups email. You can also, send them directly to me. But if you see a bird, and this includes here in Southern California, and even if it's right after the nesting season, we get a lot of birds that move from the port of LA and Venice Beach up to Malibu. That's really important. And definitely, if you are traveling and see a least tern someplace, try to get a picture that shows its leg and check to see if it's got a band. Even if you can't tell the numbers, no problem. Send it over to us. We've gotten pretty good at that, and there actually are programs that we can use that can give us a probability of what those numbers are based on photos. So it's pretty cool. The next thing is in about 2 weeks we've got a meeting coming up in Baja California, and this is open to the public. If you are interested in going, participating, please, let me know, and I can send you more information. You can also just type Alas que unen a las Californias. And again, this is going to be in Ensenada, and it's going to be a meeting mostly focused on the work that we're doing on the peninsula, but also we'll have several of the biologists from Southern California giving presentations as well. And then finally, of course, every year it's funding is an adventure. So a lot of this, we've been very fortunate to have, grants from the Griswold Family Foundation supporting us last year. But every year we have to go out and figure out how to, especially for the Mexican biologists, the biggest thing is fuel, because you have to remember salaries down there are about a quarter to a fifth what they are here, and guess what? A gallon of gas costs the same. So it's a big impact in trying to get out to these colonies. A lot of it is just transport. If you are interested in helping out, we've got groups that we're hoping to fund in Los Cabos, and then the group in southern Sonora is the other one that still needs some funding for this coming year. And so you can donate through the Ocean Foundation to Proesteros, and they're the ones that handle our funding in Mexico. So Ocean Foundation is a US 501c3. This is the link here, or you can just Google Ocean Foundation Proesteros, and it pops it in. And then this is my email. And does anybody have any questions?
Well, first of all, thank you. And if we were in person, I'm sure there would be a rousing round of applause right now. We don't have the chat enabled tonight. So what we'll ask is for folks, if you want to go ahead to ask a question, just go ahead and unmute and go ahead. We've got quite a group here, including some other biologists I can see. So would love to have your comments and questions and appreciations also for the great photography and just the excitement of this project.
Well, I'll jump in and ask a question. My name's Tom Hennebush. I'm wondering, Tom Ryan, there's a lot of Toms tonight attending, if you've determined yet whether there's any gene flow between the northern populations and the populations that you've been concentrating on down south, or are results still very preliminary?
That is what we're in the process of collecting right now, and thanks for bringing that up, because it's a really interesting topic. So, there's a couple of different possibilities for these populations, and one of them is that you have this kind of ladder effect where within a cluster there's a lot of interchange, and between adjacent clusters there's interchange, And then these adjacent clusters have gene interchange all the way from north down to south. The alternative to that, of course, is that you have isolating mechanisms in certain areas. And so one of the thoughts is that, particularly that population down in the southern end of the peninsula, and then also the population up in the upper Gulf, there's one breeds very early, and the other one breeds a little bit later than most of the rest of the population, which, that can be to some degree an isolating mechanism. And then they also both live in very different oceanic conditions. And so that's one of the things we're really interested in finding out. So Liliana is going to be doing a master's, and we've collected samples from the populations pretty much up and down the Gulf. We're going to get more samples so that we can not only look at mitochondrial DNA, but also SNPs, and really kind of try to figure out, how often or if there's gene flow, how often. And then the other is if certain gene combinations appear to be clustering. So we don't have just, random mutation all the way down. So we don't know yet. And, a lot of this, like I said, this is, it's our first year of data collection for the connectivity part of it. But what we're doing is we're taking an integrative taxonomy approach. That's what we're doing in the museums. The stable isotope paper is going to contribute to that as well, along with the DNA. And then hopefully that'll also provide kind of a basis for where to look for whether or not there are ecological differences among the population. If I had to make a guess, I would guess that the populations, if they're not connected to San Diego in the upper Gulf, and then the population down in the southern peninsula, probably are fairly distinct isolated populations.
So,
Okay, thank you.
great. So just so that everybody knows, what is stable isotope analysis and how does it work? What can it tell you?
Well, how much time do you have? No.
Sure.
So stable isotopes. The real basics of this is that when an animal is foraging, okay, it is consuming either another plant or an animal, okay? Within that area, okay, and there are unique patterns of these stable isotopes that form in the ocean. They form on land too. And so if you look at, with birds, it's the feathers. So once a bird grows a feather, it becomes inert. And so that feather reflects the ratio of isotopes of nitrogen, carbon, hydrogen, and a bunch of other things too. But the nitrogen, carbon, and hydrogen are the ones we're looking at. When that feather was formed. And depending on, again, the isoscapes, some are more unique than others. Some of these can be very similar over very broad regions. But there's a general pattern we should be able to discern if the bird was foraging near shore. So if a bird is in an estuary or near an outlet, you're going to be seeing more signatures from land plants, particularly in the carbon isotopes. And then as you get farther out into the water, those are going to disappear. And it'll just be marine phytoplankton, marine algae signatures. So that's one question: is are they on the coast? Are they pelagic? And then Also, depending on the temperature and the salinity of the water, there tends to be a change in with latitude as well. And so that's kind of what we're looking at. Trying to figure out, what these ratios are. Now the geography is tricky. That's probably the trickiest part. But what we can do is look at differences among the populations and see if they have similar patterns in their primaries. So if they have similar patterns in their primaries, that would reflect the movements that those birds made between the 2021 and the 2022 breeding season. So least terns molt all their primaries off the breeding ground. So if they don't molt on the breeding ground, it's off the breeding ground. And Larids, it seems like the average in the literature is about every 20 to 25 days or so per primary. So we don't have the data for the least terns because, well, nobody knows where they go in the winter. So we don't know where they go. We can't really look at their molt until we figure that out. So the idea is, again, it's a proxy, and it's been tried and done successfully with kittiwakes and a couple of different shearwaters. So we're trying it here with them.
Great, thank you. Anyone else? Just go ahead and unmute. We had a few comments over on Facebook. One earlier in the talk suggesting, and I'm curious to get your opinions on this, that there's perhaps some harm that is done to birds by being banded. And I'm curious what your findings are in terms of the current thinking about the impacts of banding on birds themselves.
Let's see. Yeah, that let's see. So with least terns, that's a very, very, that's a very loaded question. What I can tell you is that going back to that graphic, when the number of least terns was increasing dramatically through the 80s and 90s, that was the period when banding was at its absolute most intense. Banding was being done at Venice, at the port, large-scale banding at all of the military bases. Pretty much all of the colonies were being walked daily. The birds were being banded. The numbers were increasing. As far as mortality from banding chicks, it's extremely rare. I have never had a chick die that could be attributed to the act of capturing it and putting a band on. I've had chicks appear in bad condition before, but that again was likely I received them in that condition. There have been 60,000 bands put on least terns, plus on the west coast of California. And we have recaptured birds that are 27 years old with bands on them. I would say that my personal opinion is that the impact of banding on least terns, if it is done correctly and responsibly, is negligible. We have a lot of restrictions on us with these being endangered birds. We can't herd them. We can only pursue them short distances. We release the birds in the same area where they are captured. We limit our time in the colonies. We don't go in during poor conditions. I think that if the biologists doing the work are responsible about it, I personally have not seen anything that would indicate that it really creates any problems. And, The terns are, it's kind of interesting too with these least terns. I almost put a picture in of one that I caught. Um, and we catch them on a fairly regular basis, missing legs below the tarsal joint. Um, and about 50% of the time, um, you know, the bird has a band on the intact leg. Um, and so what we think probably is going on is they're landing on the water and something grabs them from below. Um, so that's, um, but, as far as injuries from bands and then from the alphanumerics, we do have to take alphanumerics off from time to time and replace them. And really, all we see underneath the band, we don't see abrasion. We mostly just see discoloration. Um, so it's just the leg's been out of the sun for the better part of a year. So as far as physical things, we really don't see much. And as far as the trapping goes, at this point we're only trapping when it's necessary. So if we see a metal banded bird, the only way we can find out where that bird was born is to trap the bird. But if it's an alphanumeric bird, these days we can just use the cameras, and that's effectively a recapture, and that's really minimally invasive. So all I can say is, anytime that you do any sort of research on an animal, you're going to disturb the animal to some extent. But if you ever sit and watch the Venice Least Tern colony, those birds flush, seagulls fly over, cormorants fly over, they flush like it's a peregrine falcon. These guys are pretty well adapted to dealing with disturbance in and around their colonies. So again, limiting the time, I think, is probably the most important. And then, just being aware of stress levels on the birds when you're in. But yeah, no, it's a great question though. And it's something we have to reevaluate all the time, because you don't want your activity to alter your study.
That's right. That's right. Well, thank you. Thank you for that thorough discussion there. Anyone else have questions? Like to join? So with that, I'll go ahead and end the broadcast. So thank you for everyone coming along, and we can stick around for a few minutes to converse informally. But thank you for this meeting tonight.